begin by stating your name and your role at the university?
Let's see, who am I? I think I've forgotten that. It's called memory loss. The professors aren't supposed to have that. No, I'm R. Malcolm Brown Jr. I'm a professor here of biology. I've got my undergraduate degree here at UT. I started school here in 1957 and got an undergraduate degree in 61, Hook'em Horns. And we were talking about dormitories today that were pretty familiar to what you all are staying, where you all have stayed and so forth. I came here as a pre-med student because my father was a doctor and wanted me to be a pre-med student, so, you know, I had to follow through on that, but I didn't end up becoming a doctor. It made my father rather mad, but that's the way life is. You start out what you want to do, but you never know where it's going to take you. So the point is, I was very lucky because as an undergraduate here, I got to work on some research projects as an undergraduate. One of them was they had gotten a new electron microscope here. The dean of the graduate school, Gordon Whaley, had just brought in a new electron microscope, and he let me as an undergraduate learn how to use it. I was so happy. So getting to start early on that was a lot of fun, and so I knew that I wanted to get into research. Research was so interesting, and so when I was a senior, though, I had changed my major from pre-med to botany, and that did not make my dad feel very happy. He was very disappointed. What in the world is this son doing, going to be majoring in botany? But I took a course in economic botany, which sounds kind of strange, but I learned how important plants were. I mean, you have cotton, you have rubber, you have pharmaceuticals, we have plants that we have to eat, the food chain, you name it. And so I became very interested that it's just more than collecting plants in a herbarium. So I started taking courses in botany, and when I was a senior, I started working for a professor for some cash because I was dating and I need some money. Okay, so that's what happened. I'm telling you the truth. And so I was washing dishes, and Dr. Bowles said, "Malcolm, you've done a good job washing dishes. How would you like to make culture media?" And I learned how to make culture media. "You've done a good job doing that. How would you like to transfer some algae?" "Oh, yeah, okay, I'll do that." And he said, "Malcolm, you've done a good job doing that. How would you like to go to Woods Hole?" And I said, "Woods Hole, what is that?" Well, it happened to be the world's best marine biology lab in the United States, really. And it was in Woods Hole, Massachusetts. And he had taught there. He said, "You can go there as an undergraduate student, but you'll be the only one in a graduate school class." I said, "Are you sure that I can live up to your expectations?" He said, "Yep." Well, I went there, and I just fell in love with the place. We studied algae and all the marine life, and I came back wanting to become a biologist, a cell biology specialist with algae, and that's what I did. So that's why I graduated in botany. So I changed my career in late undergraduate times, which means you all should do the same. Don't keep your careers the same all the way. You may come wanting to be pre-med, but you may find a professor or something that they do that will turn you on. And then the other thing I learned over all these years, it's not so much the university that you come to, it's the professors who teach here is the most important. What they have done in terms of science or their contributions to the academic life. And so I spent a lot of my time thinking and writing and publishing and doing research. And I think about all sorts of creative things because the administration does not tell me what to do. That's so wonderful. I can teach double courses if I want or single courses. I've always done it. Every year I've been here since 1981, I've taught a course. Never have stopped teaching. But I didn't have to because I have an endowed chair. But I just love it. And the colleagues are so nice. And of course, the most important thing is you students. And I'm very happy that you're here. It's a very special school. You made a good choice to come to a big school. When I first came here, They said, you won't like it, it's too big. Well, is it too big?
No.
But the beauty is, you can take courses from all kinds of special courses: underwater basket weaving, biology, cell biology, molecular evolution. You just name it, because we have a big staff here and a big faculty. So that brings me up to date, and I hope I haven't bored you so far with those questions.
No, sir. So, what work are you doing in your lab right now? What is your main research project at the moment? Or do you have multiple research projects?
I have multiple ones. Jack of all trades, master of none, as they would say. But basically, I've been working with cellulose biosynthesis for 30 or 40 years. We're still continuing that because there's many useful things coming out of that work. You already know that cellulose is what paper's made out of, right? This is probably paper from pulp, where they took wood and they just digested it and they ground it up and then they put it together. And so when you press it, it's hydrogen bonding between these cells. We have a bacterium that I'll show you later on that makes cellulose also, and it's much finer than this. And it's still the same chemistry. It's beta 1,4-length glucan. If you had alpha 1,4-length glucan, that would be starch. But cellulose seems simple, doesn't it? Just a monosaccharide, 1,4-length, but it takes an enzyme complex bigger than a ribosome to do it. So over the years, I've studied the enzymology of how it's made and the structure of the cellulose itself. And that's why we use things like electron microscopes. We use biochemistry. We isolate enzymes. We sequence genes. So one of my current projects is the complete genomic sequence of all the cellulose synthases in a number of bacteria that make cellulose. So that's the bacterial cellulose side. Another area that's pretty remarkable that I'm getting into right now is Mars. I'm very interested in Mars. And if the astronauts go to Mars, what's the biggest problem they're going to have going to Mars? What do you think?
Oxygen.
Yeah, there's no oxygen on Mars. There's only what? Well,
is it methane?
No, there's a little bit of methane, but there's mostly carbon dioxide, and the atmosphere is about half that, or much less than ours. So, hey, what if we could get the genes from the bacteria that makes the cellulose into a photosynthetic microbe that we could take to Mars?
Wow.
Now we could build all these things that you could not carry on your spaceship 71 million miles away. So that's a current project. That's one of my far out projects would be to get cyanobacteria, we call them. They're blue-green algae. They're very primitive organisms. You've studied them in your biology, haven't you?
Yeah.
How long do you think they've been around? What did they tell you?
What is it?
3 or 4 billion.
Yeah.
That's exactly right. They're the first organisms to emit molecular oxygen on the budding planet Earth. Before that, we were pretty bad off there. We didn't have any rusting or anything, but now that happened. And so that changed the whole evolution and phylogeny of life on Earth for sure. They're prokaryotic, meaning they're like bacteria. But you know what else they do that's beautiful? They fix nitrogen. And so if they fix nitrogen, you don't have to take lung fertilizer with you to Mars. So basically we want to get the water from the frozen ice, and it'll have to be transported over long distances. You can't carry polyvinyl chloride pipes to do that. You have to make the pipes from the cellulose, the bacteria, the cyanobacteria would provide. You can't store the water in big tanks very easily. You have to make tanks out of cellulose. So it's a structural material that is needed for prolonged living on Mars. Does that give you a little idea of how a far-out project?
Yes.
What are some challenges that you face in your research?
The biggest challenge I face in my research is getting funding to do the research. There's no doubt about it, all of us. You won't find a faculty that won't tell you that funding isn't a major difficulty. It's getting worse in this country.
Yeah.
We're spending less money on research. The National Science Foundation, the NIH, they're getting less money. If you stop and think where we put our money, I'm not going to get political, but if you think where we put our money, if we put more into research, it would just be so much more helpful. So we still need to have more money for basic research, and that's our biggest problem that I see right now.
Yeah, I completely understand. My father is in research. He works with influenza and like the vaccinations.
Really?
And everything. Yes.
Okay.
So I understand.
Is he thinking about where am I going to get my next funding? So
basically, every time I talk to him about his grants that he sends off he's always just like waiting for funding. I'm like, okay.
I've been doing that too for years and years.
But he's been doing it
I've
for
had
years.
fun. I've had grants from NIH, NSF, NASA, plus numerous other grants. But I really enjoyed that grant from NASA. That was pretty cool because the goal was to take the bacteria up into a microgravity environment. So we went down to Houston where they have the so-called vomit comet. Have you ever heard of that?
Yes.
It's an airplane that kind of dives and goes up and down, and you have about 30 seconds of microgravity, and then 30 seconds 2 times G. They invited me to go on it, but I didn't do it. That's one thing I regret. I had a chance to go on it, but I was too afraid. But anyway, we got some pretty good data, but we really needed the space station, prolonged microgravity, to really see what's happening to the biosynthesis of cellulose under micro G conditions. That affects things biophysically like crystallization, how the polymers come together. And so I didn't continue that project, but I did meet some really good people, and I have in my house my most valuable photograph is an original photograph of Earth that the astronauts made when they were coming back from the moon. You know that famous picture of the Earth? Mm-hmm. I've got one of the real original photographs. So that's the fun part. Who knows what you're going to get out of it, you know? It's a combination of research, which brings a lot of fun. You saw some of my students there. But it's an interaction with the students and research. If I can bring students, get them to appreciate science through research, I think I've done my job. And the only way I know is many years after they leave, they'll write me or call me, hey, I'm doing this, you know? Did I answer that a little bit?
Yes, sir.
All right.
What advice do you have for students who are interested in your field of work, in your subject area?
Come visit me. I mean, I would have never been in what I'm in if I hadn't met that professor and started washing dishes and got involved. I have been teaching undergraduates research long before the freshman initiative. That was a very late thing. I've been here much longer than that, and always believed that undergraduates could do the research as well as graduate students. Just a matter of meeting me at the right time, and you know. But now they went from the juniors and seniors through my coursework now to freshmen, which at first I thought, oh no, wait, this is not going to work. I mean, here they are, fresh out of high school. What will they really know? What have they done? They're not going to get the core courses of organic chemistry and math and physics that they need to really make the most of biology. Guess what? I was wrong. This freshman initiative has turned out beautiful numbers of students, and they now take my courses as you saw. A couple of those students that you saw earlier, they came to the freshman initiative. So that's good. You're lucky to have that here, you know?
Yes, sir. And as you know, we're in, the freshman bio, intro bio. How does your work relate back to the core of biology?
Oh, well, the most abundant macromolecule on Earth is cellulose. 10 to the 11th power tons made and destroyed annually. So besides chlorophyll and biomass, it's the next most abundant material. So I work with something that humanity needs for life. So that's why it's important. So the more we can learn about it, if we can produce it in larger quantities, if I can get trees to grow faster or to be stronger, we can help on a large scale. For example, I spent 3 years putting some algae out at the Brackenridge Field Tract out there on our campus in Austin, and we grew algae outdoors in big tanks, and we got them to produce beautiful amounts of biomass. And guess what? enough biomass that we could probably have converted that into at least 10 to 15% of all of our automobile fuels, just from growing it in West Texas, near Midland or Odessa, in ponds next to the oil fields of the 2 million acres that the University of Texas owns. That's the kind of thing you could do. But I couldn't get anyone interested. Now came along something called fracking that shifted the emphasis away from sustainable energy, because we still have more. But we do need to think about fracking. We need to not think about fracking. We need to think about continuing about the biofuels.
Is it an issue of money?
Oh yes. Oh yeah. It's a terrible issue of money.
So what's a potential solution to that?
Oh, if we could get enough R&D done to build a new cellulose synthesizing organism in West Texas on university lands where nothing else grows, we're not replacing trees, we're not replacing corn or cotton, we'll have a new crop. Think of the economic importance of that. It'll cost money though to develop and scale up how to do that, because you have to use microbiology, you have to use fermentation technologies, all these new things, and that's part of it. But it's feasible. I think very far out. But if I didn't do that, and if many of my colleagues didn't do that, we would just kind of get into a doldrum of just let's teach, let's just show the regular things. I mean, computer science is so cool. Look at this. This is my Apple 4S, okay? I love this, okay? Even though I had a Radio Shack earlier, this is one of the most beautiful things. And I always admire Steve Jobs for not only producing a superb computer, but making it beautiful on top. Someone told me that he was doing scripting or something like that when he was in his early life, and that gave him the idea to make good type. I got the first Apple computer they made, and that's the one that we had a GUI where you could see all the normal typeface. Computer science has come a long way. Now here's something that's kind of interesting, since some of you are in computer science. That and biology together will be great partners, because we need more and more approach to understanding complex things like phylogeny and the origin of life on Earth. This is one of my things I want to do. I want to understand, where did life come from? There are some prevailing views about it. I have a very different view about where life came from. I don't think it came from Earth originally. If someone's going to hear this, that's okay. I'm willing to have them say it. I think life came from another part of our solar system. I think it may have originated from something like Mars or the moons of Jupiter or Saturn. Why? Because you have these carbonaceous chondrites that migrate through space. They have a lot of complex organic molecules. So what if there were bacteria already made on Mars and they got transported to Earth when all these conditions were just right, and then suddenly it's like a test tube where you put E. coli in. You'd have explosive growth. And that's what happened when life first originated here on Earth. I have a hard time thinking about, how did you form a first cell? How did you get lipids going? How did you get DNA, a molecule, to replicate so perfectly? I think it took an awful lot of steps to do that. Now someone's going to say, oh well, okay, didn't come from Earth. Well, how did you do it on Mars? Yeah, they'll ask that question too. You guys, guess what? You're going to get to explore and answer these questions. I think that's the most exciting thing you could ever imagine. I think astronomy, cosmology is really a way to go. And I'm very interested in a new field of science. It's called astrobiology. Have you ever heard of that? Oh, mark my words. That's studying the origin of life and our solar system and our planets and our universe. How do you think we might be able to detect life elsewhere in the universe? Have we made any attempts to do so?
We've sent radio signals out, but there's no guarantees that other life forms have radio signals they could send back or even receive. That's right.
That's called Project SETI. Have you ever heard that? Search for Extraterrestrial Intelligence. It's near San Francisco, and Have any of you ever seen the movie called Contact? Oh, please
It's
go
another
see it.
movie. What was the first one? Power.
Power to 10.
Power to 10.
But that's kind of a little science thing. But this is a real Hollywood movie, Contact, starring Jodie Foster. Please go see that movie or get a video. It's the most important, significant movie you will ever see as a biologist right now. And I'm not going to say any more, but you have to tell me what you thought about it after you saw it. It was an earth-changing thing for me. So here's a professor who's talking about contact and things on Mars. I'm happy to show you around the lab. This room here represents our electron microscope. I'm very proud to say, if you can see it, this particular instrument can magnify from 45 times to 30 million times. So we have no trouble seeing single carbon atoms. And we use very sophisticated computer programs to look at this. We work in the so-called spatial domain, which is you create an image with an electron beam like you would a light microscope, but you punch the diffraction button here and you go to the frequency domain, which is like Watson and Crick. We're looking at DNA. They just line them up and hit them with X-rays, and then you got the diffraction pattern from the frequency domain. At that time, math was very hard to go by, and they finally found out how to get an image of what it was like. Guess what we can do now? We can image the double helix of DNA, do a fast Fourier transform, and convert it back into the Watson-Crick reflections that they got. So that's going around 360 degrees. So much fun to do things like that. This little O-ring right here is all there is between 10 to the minus 7th torr and atmosphere. They're working so hard now. I saw you jump. Well, when the first student does it, I don't tell them about it. They really have a fit. And that just turns on the mechanical pump, and it sucks it in there, and it's pumping this now out to 10 to the minus 2 torr. Then we'll let it suck in a little bit of air into the big high vacuum. But right now I'll let it do that. So that's the way you can take things in and out of the scope. Okay, now I can open it. Now I can open this up. Go in there, and that's it. And oh, this is a beautiful scope. It really is. I'm so lucky to have it. What makes our lab kind of different than all the other labs on campus? We have the very advanced molecular biology lab, very advanced biochemistry lab. We do all that, and we have, in addition, very advanced microscopy lab. Not only this, but also light microscopy. I'm going to show you the best light microscopes in the world. These are some of the best light microscopes in the world. This is about $100,000 when I came here in 1981.
And now?
$250,000 maybe. But we have really expensive cameras, like that's a 25 grand 3-chip CCD camera. So we keep making them modern to access the imaging capabilities. Mm-hmm. I started out imaging cameras on things like this.
Oh my goodness.
$450 for 1 hour's recording. That's what it used to cost. We've come a long ways, haven't we?
Yes.
And so I have different kinds of light microscopes. This is a compound microscope. This is a dissecting scope. This is an inverted compound microscope. They're in special rooms that have HEPA filters, so there's no dust in here ever bothering them. And they have Faraday cages around them. It's copper so that no interference takes place. Then I had them pour 3 feet of concrete here to keep things stable. And we don't have any problem with stability. We needed that for the electron microscope because, oh, I forgot to show you. I wished I could have shown you when I had that at high mag, when I whistled, sample would just vibrate. So wow. It's amazing how vibrations will cause things to, not be stable. Well, and then we use standard computer programs here, and I use a lot of Adobe Premiere and all that. We do time-lapse videography, and we can do time-lapse electron microscopy. So come back sometime and we'll show you some really beautiful things here. I can move this camera over on here. You can get to see things. This is really a beautiful scope. I spent a year in Germany at the Zeiss factory. That's where I learned my microscopy. I'm so lucky to have done that, you know. And look behind you at all the different kinds of objectives. You would be amazed at what we have. We have bright field, dark field, fluorescence, Yamin-Lebedev dual beam interference, polarization microscopy. We have all these different kinds that people don't even know about hardly. But that gives us a handle on cells. We can see individual processes in cells that you would never believe you'd see. Well, let's go show you where we grow some of these algae and bugs, okay? Microorganisms. So we have culture media, we have test tubes, we have agar, we sterilize media. We have a very large autoclave room right over here. After we autoclave and sterilize it, we have these rooms in here where we transfer cultures. And these are laminar flow hoods. You can't see much in there, but you can see the autoclaves in there. And then after we get the cultures in their vessels, we grow them in special culture rooms. I'm going to let you come in here and see more. Come on up through here. This is where we grow the algae. Come on in. Okay, these are some marine algae. See them right here?
Oh, wow.
Now, what I want to show you that you haven't seen, I want to show you the world's largest cells. They're so big you can see them with your naked eye. Go up close on them. See those little round, little spherical things? Those are giant multinucleate cells that normally grow in the ocean, and they make beautiful cell walls of cellulose. That's why I study them. But let me see. I have a much better one I can show you. Oh, here's some pretty nice ones. These are pretty big cells. These ones that are white, the cells are dead. But you can see them with your naked eye. Most cells you cannot, right? Have you ever heard of cyanobacteria, blue-green algae? I told you that, didn't I?
Yeah.
Here's some cyanobacteria here that we're growing right here. So this room is, we have 4 rooms like this. This one has to have light for photosynthetic microbes, and that's what we use it for. Then we have another room that we grow the bacteria in that don't require photosynthesis. So I'm going to show you that. Then I'm going to take you to my office and show you the actual cellulose that these bacteria make. It'll blow your mind. This room is where we grow our bacterial cellulose, and they're made by bacteria. Here's a living culture, and if you see that little biofilm on top, that represents some bacteria and their extracellular product cellulose. And the shape of that cellulose is the shape of the tube. It gets thicker and thicker with age. So here's a young tube, cellulose. Here's an older tube. It's that thick. So if I take the same thing, culture medium, and put it in these giant trays up here, I can produce a huge sheet of cellulose. And it's the strongest cellulose in the world. And I'll show it to you. You may be interested to know that we're making, going to make a violin that will be comparable to the Stradivarius violin using the cellulose. We have all kinds of projects like that. Oh yes, oh yes. It makes the world's best burn bandage. It stops pain instantly. And we're interested in things like that. But this cellulose is very special. But I want to show you where we grow it in this room. Again, it has HEPA filters, sterile. We're not hurting anything because you're not going to stir up the dust. But here's another one that's pretty nice if you want to. That's a nice little culture right there. One good thing about this cellulose and these bacteria, they're gram-negative bacteria, but they're non-pathogenic, thank goodness. Have any of you ever eaten any kombucha?
Yeah.
That's exactly what this is. This is the same bacterium that makes kombucha. Well, I've got two rooms, and what I'd like to show you is some of this cellulose. You saw it in the test tube in the lab. Here's a piece of it that was made in a membrane in one of those shell things. This is the world's strongest cellulose. Wow. It's so strong you could build armored cars out of it. You could build airplane wings parts. You could build rocket casings. And it's all digestible if you use the cellulase enzyme. Here, take this and let's have a little contest. Okay. Are you going to pull?
I'm going to pull.
Oh gosh,
You're
strong. But this is a piece of membrane, but this is a thin one. When you make it, when it's made, you saw the gel in the tube?
Yeah.
Mm-hmm.
This is what the gel looks like in a tray. It's a big old bowl filled membrane. It holds a lot of water. That's something.
Wow.
And then if you really take a thick one like this and dry it down, that's what it looks like. It's pretty thick.
Looks like a cracker.
Yeah.
Saltine.
Yeah. What I want to show you, let's see if I can find it. Oh, if you dry it down on something like this, it produces a dried cellulose that has the same shape and feeling. You can control the parameters of it, you know?
Wow. So, I guess essentially you can make it into whatever you want to, mold it into whatever you
can
want.
mold it. And I invented and have a patent on it of making a non-woven project. And it was kind of a simple little lab experiment that led to this patent and this invention. Instead of putting the culture medium in those test tubes, I put it into an ordinary laboratory glove. And then I inoculated it with the cells, and guess what they did? They made a cellulosic membrane that was not a flat one, but the same shape as the glove. Because they need oxygen to make the cellulose, so the glove was oxygen permeable. So we made a non-woven glove. That means I can now make tubes. These tubes can be used to replace arteries and veins in cardiac surgery. You can build all kinds of special complex geometric objects, and that's what we do. And then I'm going to show you something else. We actually can put carbon nanotubes and other carbon components in the culture medium, and the bacteria take up the carbon with the cellulose, and this is now an electrically conductive membrane. So this can become the world's best battery. We're on the road to making ultracapacitors and batteries out of this. And here I'm going to show you, I'm always proud to show this to anyone. This is the world's thinnest paper. You want to see how thin it is? Yes. It's so thin that I have to be careful. Let's just see if I can get a piece of it out here. Well, I'm going to try this right here. This is, okay, here it is. It's pretty thin. You see how thin it is right here?
Does it rip easily?
Well, I mean, I
Yeah.
ripped a little bit of it off there, but I want to show you something. Look how thin it is. Let's see if I can.
Oh my god.
Oh my goodness.
It's a helicopter, isn't it? I've never made a helicopter before out of it. Okay, how thin is this membrane? Give me how thin is it?
Oof.
I'm going to take this off now.
Less than a nanometer, or is it
Oh,
about
no,
a
no,
nanometer?
nanometer is really thin. I mean,
Oh, okay.
well,
A third of a millimeter?
that's a good way. How many nanometers are in a third of a millimeter? Let's
Oh
think
god,
about
I could
that.
do that.
Well, that would be 300 microns. Yeah, 10,000 nanometers, 1,000 nanometers per micron. So that's a lot of nanometers. This stuff is 100 nanometers thick. It's 1/10 of 1 micron, and a red blood cell is 6 microns. So
What is a normal sheet of paper?
Oh goodness, 20 microns, 50 microns. I want to see this. I want you to see this one float. Let's see if it's going to go. Oh.
That's so cool.
I need to have that back. Can someone recover it for me?
It'll come out.
I got it.
Oh, good job.
Thank you very much. Well, so you've seen the world's thinnest paper.
And so that's the same thing, just...
It's no different than that. It's cellulose. It's just that
Yeah.
we grew that membrane for about 1 hour instead of 4 or 5 days like this. Then I've invented something that I'm really proud of. And the basis of it is right here. Well, this has got some marks on it, but see how this is the same cellulose, but look how beautifully fine it is. These you'd have to have an electron microscope to see the structure. You couldn't see them with a light microscope. So this is the basis for my invention called electronic paper. In other words, I can put electrochromic dyes into the cellulose, and when these receive electrons, they absorb light. So if I build an electronic backplane into this paper, I can build a pixel generating system that creates an image. In other words, I can build the world's finest imaging system on a piece of paper, much better than a computer screen. Even Steve Jobs would be jealous. But the idea is to actually, it's reflected light rather than transmitted light. And so that's just one of the applications. Here's what I'm trying to learn right now about how violins are made, because we're working on that. We have guitars over here. I'm going to try to improve the sound quality of guitars. I went to the Austin Guitar Society's concert the other night. Did you ever go to that over there? The Russian guitarist that played beautifully. My goodness. The problem is it had to be mic'd up. You know, guitars just don't make that kind of... I'm a classical pianist myself. I'm a composer. And so I just have to have things like that going. Yeah, my CDs are all over the world.
Oh, that's what my grandma was telling me.
Yeah.
Did you play in the NFL also?
Yeah.
I would have had so many brain injuries. I don't think I could. No,
That's very true.
I do worry about football players. Wouldn't it be nice when you think about this? It's a wonderful game, a wonderful sport. Why can't they design some things that are less harmful for the players? I mean, why do we want to go out there and just let people get concussions and have brain damage from them? I guess you're putting this on here, but I'm just saying, I think we should be careful. Here's something else that we're making out of it. Musical instruments. Yay! This is the same.
Oh, wow.
The eyes, the X's are. Okay, you guys. Oh, well, I'll show you before I go. This is what cellulose looks like. This is my baby. That's what I've been studying for all these years. It consists of a simple glucose back monomer, and they're linked by an oxygen bridge here. So that's glycosidigly linked beta 1,4. And then on one side there's mostly van der Waals forces that would hold the chains together. On the other side there's hydrogen bonding. And so that's all the complications of what we study. I'm not going to go into details, but that's the basis on making this crystalline structure that we now see that is known as cellulose. So I hope this has been a good chance to tell you all about it. I'm so proud of you all as freshmen to want to learn more about it. And hello, Sata. How are you? They came to me.